Hello, everyone. Welcome to today's webinar, Characterizing a Flow Cytometer's Potential for Small Particle Detection. I am your moderator today, Joanne Lannigan, and today I have the pleasure of introducing the two webinar speakers who are well known to me as colleagues and as friends and as EV experts. And so I think it's very exciting that we have both of them here today. I did want to tell you that at the end, if you have some question and answers, if you could please put them in the chat box and we will get to them before the end of the webinar. Thank you. And I'd like to first introduce our speakers, Vera Tang and Joshua Welsh. Take it, guys.
Welcome to our webinar today about small particle detection. This was a tutorial that was given at Cyto 2024 in Edinburgh on May the 4th. The title of our webinar is Characterizing a Flow Cytometer's Potential, and we'd like you to know that this is a method that is instrument agnostic. So that means it should work with pretty much all of the instruments, standard instruments that you would find in your flow lab. I'm Vera Tang. I'm the operations manager and adjunct professor at the University of Ottawa in Ottawa, Ontario, Canada. I run the flow cytometry and virometry core facility, and I'm also an ISAC SRL emerging leader, and I graduated in 2023.
My name is Joshua Welsh. I'm a staff scientist in the Advanced Technology Group at BD BioSciences in San Jose, California. I'm also an ISAC Mary Lou Ingram Scholar, and I graduated in 2024. So just some quick disclosures. I'm an employee of BD BioSciences, and I'm also the owner of the Measuring Stick Limited in the UK, which is the distributor of the FCM PASS software that's within this presentation.
I am an employee of the University of Ottawa, and I also consult for Beckman Coulter currently. All right, so moving on. Our tutorial objectives today, we have four main ones. We're going to go through a basic introduction, outlining what are some of the challenges that face small particle detection, and why do we want to use flow cytometry for small particle detection? We'll go through some key concepts, mainly definitions and concepts that govern small particle detection by flow cytometry. We will also introduce you to a workflow, which we have worked out over the years, how to optimize, quantify, compare, and maintain instrument performance for your small particle flow cytometry. And last but not least, we'll show you what all of this can do in an advanced analysis mode. And that means we are going to be deriving epitope number, diameter, and also refractive index. So let's start with our first tutorial objective, which is to give an introduction on what some of the challenges are and why we want to use flow cytometry for detection of small particles. Because we know that flow cytometry works very well for cells. Why do we want to use it to detect these small particles? So how small is small? When we're talking about small particles, we are essentially dealing with biological particles such as extracellular vesicles or EVs, viruses, both mammalian and plant and insect, and also marine viruses as well as phytoplankton and some small bacteria vesicles as well. These are generally under the size range of 1 micron down to, I would say, about 40 nanometers, which is the approximate realm of the particles that we are dealing with.
So this slide shows some commonly used analysis methods, and they're all analyzing the same sample of dendritic cell-derived EVs. And these have been isolated with a QEV column, and you can see the isolation fractions there from 5 to 8. You can see the diameter distribution measurements, even though these are the same samples, are fairly different between characterization methods. This is mainly due to limits of detection. You can see that transmission electron microscopy, which isn't really limited in its lower limit of detection for an EV sample, can see a bimodal distribution. And with the microfluidic resistive pulse sensing, you can see that part of that lower distribution is cut off due to the lower limit of detection. You can also see that flow cytometry is cut off with its lower limit of detection being a bit higher than the microfluidic resistive pulse sensing, but the data is concordant between them. Something to note here is that the three methods I've spoken about so far have been fairly precise in their measurements, and if I overlaid all of these plots, that larger of the two peaks would be overlapping with one another. One of the issues here with nanoparticle tracking analysis is the lack of precision in its diameter distribution. And this is why you commonly get a single distribution curve that's log normal. And it's kind of a smear from around 100 nanometers, which tends to be its limit of detection. This just really highlights why precision is needed in our measurements and how we can get different diameter distributions from the same techniques. So a nice study was done a couple of years ago looking at the concentration, the reported concentration of platelet vesicles in publications over time. And what was noticed by the group is that it's increasing. So the number of vesicles in our blood isn't increasing over time. What's been changing is the rigor and reproducibility in the methods that we're using, and also the sensitivity in the techniques. So this inherently just means that our method of reporting concentration is flawed. And really what we should be talking about is the particle concentration detected above a set limit of detection. And in this way, we would have reproducible measurements over time, irrespective of the sensitivity changes that are inevitably going to occur as technology advances.
So this brings us to some key concepts and definitions governing the detection of small particles. In the previous slide, Josh talked about the limit of detection, and this is definitely one of these very, very important concepts that we need to understand and how it should apply to small particles. The limit of detection essentially determines what you can see. So in this image here, we have battlegrounds between the good and the bad. We've got Chewbacca and we've got Jar Jar Binks against the evil empire. We've got Darth Vader, Captain Phantasma. Looks like a pretty...
We've lost audio again.
Sometimes what you see is not what's actually there, because maybe your limit of detection or your ability to detect is not there, but those particles are still there. So how do we determine this limit of detection, and how do we define it? And this is where calibration comes in. So what is calibration and why is it important? Calibration converts arbitrary units into standard units of measure. So in this case, we can actually define the limit of detection as a standard unit of measure so that it can be compared across different platforms.
In flow cytometry, there are lots of standard units, and they are split depending on whether it's a fluorescent parameter or a light scatter parameter. With fluorescence, we have molecules of equivalent soluble fluorophore, equivalent reference fluorophore, and antibody binding capacity. Whereas with light scatter, we can convert the arbitrary units to units of diameter or scattering cross-section. So not all of these units are equal. So in the case of fluorescence units, the antibody binding capacity and MESF units are hardware agnostic because they relate to a single fluorophore. Whereas if you're using another fluorophore, say a broad spectrum fluorophore on a rainbow bead, and then convert it into units of FITC, that broad spectrum fluorophore won't respond the same as FITC under different illumination conditions. And that means that calibration is hardware dependent. And this can cause issues. So ideally, you want to stick with hardware agnostic units where possible. So fluorescence calibration is fairly easy to do. You will typically buy a set of beads from a manufacturer that have defined numbers of molecules on their surface. You will acquire those on a flow cytometer, and with the detected MFIs, you will plot them against the assigned units on the side of the bottle and draw a regression line. You will then use that regression to convert all of your data into standard units. One of the issues with fluorescence calibration using MESF and antibody binding capacities is they tend to be a little bit more expensive than buying earth beads, which are typically a set of rainbow beads. Something that you can do to get your money to go further is you can buy a set of hardware agnostic calibration reference materials like MESF or ABC beads, and then you can cross-calibrate the broad spectrum beads, like your 8-peak or your 6-peak beads, using that regression line as you would with normal cell data. This would create earth units again, but those earth units would be specific for your flow cytometer, and therefore they wouldn't have the same limitations as broad spectrum beads that you buy and were calibrated on someone else's flow cytometer. So we see that when we use fluorescence calibration, at the top of this plot is a virus, the murine leukemia virus, and it was acquired on 2 different flow cytometers, which are the black and the blue lines, and then 3 different settings on those 2 cytometers. So you can see with uncalibrated data, we have 6 populations, even though it was the same sample at the different settings and on the same cytometers. Whereas if we calibrate that data to the number of PE molecules that were stained on that virus sample, you can see that now all of those populations are overlapping. So we've removed the hardware-dependent variability, which is a great way to report data because it's now agnostic of the instrument platform. How this comes into play with concentration is really important. As we said at the start, not all flow cytometers have the same sensitivities, and we aren't detecting all of the EVs. So when we gate around a population and report concentration, we're actually reporting a concentration within an upper and a lower limit of detection. And you can see here that we acquired the murine leukemia virus on 2 different flow cytometers. On this first flow cytometer, in plot E, you can see that part of that population is being cut off. And that has resulted in a lower concentration than the cytometer on the right. And now if I show you the red box where we account for the limit of detection, those concentrations now are more concordant with one another. So light scatter calibration is a little bit more complex than fluorescent calibration. The main reason for this is because it's non-linear and it's dependent on the collection angle of the system. The collection angle of the system is different between flow cytometers because they have different cuvette geometries, and it can even be different between flow cytometers of the same platform just because the lasers and their relative position can change.
Historically, we see a lot of publications that utilize these sizing beads to determine a gate to which they will then utilize on their EV data. Now, there's a problem to that because, as Josh was mentioning, as a result of the differences in collection angles, these beads and also the materials that the beads are made of, these beads will look different or very different from your EVs. Here in these slides, what you're seeing is how the polystyrene beads adhere when collected on side scatter, and once they are calibrated, how they look like in terms of diameter. So also we are comparing the differences of materials, so silica beads versus polystyrene beads. As you can see, these beads don't really reflect the size of your EV samples.
So this slide shows some recombinant EVs. These are commercially available, and we're going to use these throughout the rest of the presentation. And they're a great reference material for you to use on your own flow cytometers. This shows uncalibrated data, and you can see that between the Cytoflex and the Aurora that they were acquired on, that the populations don't overlap. We just gave you how light scatter and fluorescence calibration was done. And now when we switch to the calibrated data, you can see that the data is completely concordant, irrespective of these 2 instruments, which are very different in their collection angles and their illumination optics. One of them is a conventional cytometer, one of them is a spectral cytometer. So this is just a really good example of why calibration is so useful to have platform agnostic data.
So hopefully from what we've shown you so far, you are excited to know how we've done this calibration and what is involved. Our next objective is to go through the workflow. We'll show you how to optimize, quantify, compare, and maintain instrument performance for small particle flow cytometry. So what is this workflow that we keep talking about? This workflow was published in 2023, and it is essentially a quantitative flow cytometry method that enables end-to-end optimization of cross-platform extracellular vesicle and small particle studies. Importantly, this is an instrument and assay agnostic pipeline. So it means that it should work with all of your standard flow cytometers in your flow lab. It is, as I was saying, used to detect all small particles, not just EVs. So viruses, very small bacteria, bacteria-derived particles, and EVs. And it is applicable to both fluorescence and scatter triggering methods. And what is the most important thing for you to know is that this pipeline will help you calibrate your data, and it will also help you derive the most sensitive settings for your flow cytometer. So this method utilizes a software that was developed by Josh and is currently being distributed by the Measuring Stick. It's called FCM Pass. It is free for academic use, but unfortunately not for those of you in industry. So this workflow is laid out in the first figure. We go through a series of steps that allow us to determine if a flow cytometer is able to detect small particles. The first of these steps involves cleaning and QC. This is something that anyone who is used to using a flow cytometer will be familiar with. This is essentially your manufacturer recommended cleanings. For our Beckman Coulter Cytoflexes, this is a deep clean. Cytoflex Aurora does a clean flow cell function, and BD Fortessas are your long clean functions. Manufacturer recommended QCs for Cytoflex, this would be your Site Experts Daily QC. Cytec Auroras would be your Spectral Flow QC. And of course, for your BD instruments, this is your CSNT. The next step is to optimize your side scatter detector settings. For side scatter detector optimization, you will need some materials. We recommend using a fluorescent 100 or 200 nanometer polystyrene bead. We call this method scatter detector setting incrementation. And the reason why we wanted fluorescence speeds here is that we want to trigger on that fluorescence in order to run the full range of voltages and gains in your scatter.
So one of the unique things about this methodology is that we are using the system optoelectronic noise floor. And you can see that in the top left population in the blue. So this is a unique population that you will all have, and it's kind of subsampling the baseline, just the very tip of it. And it's really important to know your noise floor. It's also important, as Vera said, to clean your instrument so you're actually looking at the true noise floor rather than just debris or dirt in your system. And the reason that we want to use a fluorescent trigger when we're doing a light scatter calibration is we want to sample the noise equally between all of the different detector settings. If we used a light scatter trigger and then we increased the light scatter detector settings, the noise would move, and then we'd have to move the threshold and the noise and try and equal them out every time, which is not going to be done very accurately. One of the other things we do is once we've acquired all of that data, we normalize it. So the intensity of the single bead population we make equal, and then we see how the noise relative to that population decreases. So what we're essentially seeing in that first plot is the signal to noise ratio changing. When we look at the noise in this furthest right plot where I've taken the 95th and 99th percentile of the noise, now we're looking at our lower limit of detection. And we obviously, when we're looking at small particles, want to use the lower limit of detection. That is the smallest, where we have the highest sensitivity. And in this case is around a gain of 2,500.
Once you have determined your optimal detector settings for scatter, it is now time to determine a threshold. So to do this, we usually recommend the use of clean PBS or buffer, and also clean tubes. Generally, the PBS that we recommend is the commercial sealed bottles of tissue culture grade PBS. The method is very straightforward. You're going to take your settings that you determined previously, and you're going to run your clean buffer on the lowest flow rate. Now, for most standard conventional cytometers, this is about 10 to 15 microliters per minute. You're going to adjust that threshold until the rate of events per second lies somewhere between 1,000 to 1,500 events per second, and that will be your trigger threshold on that optimal detector setting. Note that if you are attempting to determine a fluorescence trigger threshold, you will first have to optimize your fluorescence detector settings. Now, the next step outlined in our flow chart is to check whether that event rate is stable. So you've determined your trigger threshold and you're about 1,000 events per second. And you want to make sure that when you're running this PBS at the consistent flow rate, that you're seeing about 1,000 events. So if you do see unstable events, this likely means that you will need to do more cleaning. So for those of you who are asking, what does unstable versus stable look like? This is an example. This is an example on the Cytoflex. If you see the events, there are some spurts, some little, I call them event spurts, as opposed to your stable on the right-hand side. So what you want to see is the image on the right-hand side. And if you see something like what you see here on the left-hand side, then you do need to go back and clean some more. So the next step in our workflow is to optimize our fluorescence detector settings. The materials that we utilize here are multi-peak rainbow beads, and the method, similar to with scatter optimization, is an incrementation of the detector settings. So in order to use these beads, we have to trigger on scatter since we are going to be changing the fluorescence detector settings. So we run the beads for the full range of voltage available for fluorescence detectors, and we record each of these as a separate file.
So fluorescent detector optimization is commonly done now. And one of the typical ways is often called a voltration. You'll take some multi-peak rainbow beads and you'll measure them across a lot of different detector settings. And then the stain index or the MFIs are looked up in some way. So looking at the stain index, the populations are barcoded. So the red is removed because everything is being compared, the separation from the first peak. And you can see that the optimal detector settings, if I was just going to use these multi-peak rainbow beads to optimize the detector settings, would be around 800. So if we move to the next slide and we introduce this noise floor as we did for the light scatter detector setting optimization, and we look at the separation of those multi-peak rainbow beads from the noise, you'll see that now actually the optimal settings are around 2,500. And the reason for this is because the dimmest rainbow peak actually has a huge amount of autofluorescence. And that's why it's not a true background for the system. And to get a true background, we need to sample this noise floor. So fluorescent calibration, as we spoke about, will just require that you have some MESF, EARTH, or ABC beads, and you're going to acquire these at the same settings as your optimized detector settings that were determined in the last few slides. So the way you can do this is fluorescence calibration is semi-automated in FCM-Pass and will write all of your FCS files to calibrated units. Or if you do this in an analysis software, FlowJo, FCS Express, and even in Excel can all facilitate doing regressions and applying them to statistics. So light scatter calibration isn't currently facilitated by most conventional flow cytometry analysis packages. To facilitate it, you'll need some NIST traceable size standards, and it's really critical that you use these and not fluorescent beads such as fluorospheres or megamix beads, because they're not size standards, they're not homogeneous, and they don't have a single refractive index. Because they're fluorescent, they have a complex refractive index. And it's really important for softwares like FCM-Pass that it can derive the collection angle. For Rosetta calibration, it actually only works with the beads that they sell, and those are NIST traceable calibration beads as well. So the reason that this isn't easy to do is because it's nonlinear, and you'll see in the next slide, as we've spoken about earlier, that the light scatter calibration beads and that these curves will all be unique due to the refractive indices and the collection optics of the flow cytometers, which all need to be derived from those calibration standards.
And just something to add, the practicality of calibration means for those of you who run a core, you know exactly the limit of your detection on the instrument, down to the types of particles that people are interested in looking at. I know I have a lot of people that come to my core and they say, can your flow cytometer detect EVs that are a certain size? And I can confidently tell them, yes, my limit of detection is, for example, 80 nanometers for EVs and 60 nanometers for polystyrene. So the next step in our workflow is to assess that instrument sensitivity, basically running the particles that maybe you are familiar with, that you consistently use in your lab. I like to detect, I like to use some reference materials. For me, those are viruses. Some of them are recombinant EVs, like Josh was mentioning in our previous slides. These are commercially available and consistent, so that you can purchase them over time. Any number of these materials you should be able to run on your cytometer and maybe reference to literature to know whether these should be detectable on your particular platform. And if they aren't, then you can perhaps go back to the manufacturer and get some engineers to help you perhaps tweak the alignment on the instrument.
So as Vera said, having controls, positive controls, are really useful, and you want to validate your optimal settings. And this is an example of performing those detector setting incrementations. And then every time we do them, we didn't analyze just the beads that we were performing the optimization with. We actually analyzed some positive controls as well. And you can see here, if we compare the PBS control to our EVs, how the limit of detection changes for light scatter in the left column and for fluorescence in the right-hand column. And it's very clear that the detector settings have a huge impact. We go from kind of cell detector settings, we have a limit of detection at maybe 120 nanometers roughly. And then when we optimize the settings for EVs, we go down to a limit of around 81 nanometers. And for fluorescence, this is an even bigger improvement in terms of units, where we've gone from gains of probably 200 or 300, where we're in the hundreds of ERF, down to just 42 ERF at optimal detector settings. And you'll see that the output graphs that you get with FCMPass will show you the lower limits of detection in calibrated units, and you can go away and validate these with EVs. The other thing to add here is that these optimizations are not only just for detector settings. If you have other handles that you can pull, like the laser powers, you can also optimize your laser powers and know which laser power is the best for the particular fluorophores you're going to use. And it won't just be a laser power is the best for all fluorophores. It will be fluorophore dependent. And the window extension is also another criteria that really shows a big impact. So really, we should report all of our data using the area statistic, because it's less biased to outliers, especially when you approach the lower limit of detection. And what a lot of the earlier papers would recommend is to use height, because they didn't see this. What really wasn't addressed in a lot of those manuscripts is that they were still using the default window extensions, which tend to be kind of a couple of microseconds, usually 5 to 10 microseconds. And that will really impact your detection limit with the area, because you're adding on a bunch of time for the particles to integrate, which just adds noise to your signal. And you'll see that when the area window extension is set to 0, you should always have a higher sensitivity when compared to the pulse height.
So this brings us to the last step of our workflow, which is to begin your experiment. So at this stage, you will need to refer to the current frameworks and guidelines for EV and small particle flow cytometry. The ones that we have listed here, they do say EV, but they do apply to other small particles such as viruses as well. We have the MyCEV guidelines to outline sample handling, MyFlowCyte framework, which is cytometer specific, and also the MyFlowCyte EV framework, which is for small particle cytometer specific guidelines.
So one of the key reporting frameworks for small particle flow cytometry is MyFlowCyt EV. And this is an extension of the cellular standardization and reporting practices paper MyFlowCyt. And it came around from a group of researchers spread internationally between the societies of the extracellular vesicles, the advancements of cytometry and thrombosis and hemostasis, that were self-proclaimed gurus with small particle analysis. Some EV samples were sent between all of the sites, and then the data was aggregated. And we realized that none of the data was concordant, and we didn't really know what we were looking at. And what we ended up having to do to really understand how each of us was using our flow cytometers for EV analysis is to construct a reporting framework so that we knew how to replicate one another's results. And the result of this is MyFlowCyt EV. So if we look at this table, it's split into 7 broad categories. The first 2 are around reproducibility, which are the pre-analytical variables and experimental design. So, how did you get your sample? How was it isolated? You'll then have your sample preparation with respect to flow cytometry. So how was it stained? And then we'll move on to assay controls. So these are going to be proving that you're actually looking at the thing you're looking at, whether that's a vesicle or a virus. We'll then move on to instrument calibration, data acquisition, and characterization. So these are forms of standardization where we're going to try and make the data agnostic of the flow cytometer and remove that hardware dependence so that we can make comparisons over time or between flow cytometers. And then we move on to reporting that data. So how did you gate your data? How did you calibrate it? And then how did you gate it? And then finally, one of the really critical parts is transparency with sharing your data and making it available, because it's very difficult to share all of the plots and quality controls that you need in your manuscript or supplementary. Sharing, there's really no substitute for the FCS file itself. So we're just going to focus here on some of the key assay controls, not all of them. And the first one is coincidence detection.
So what is coincidence? This is something that you may have heard of. Most people who have heard of some talks on EVs have heard of the term coincidence. They may have heard of the term swarm. So exactly is coincidence? So coincidence is multiple particles present within the interrogation zone at the same time. So if you look at this figure on the right here, you see cells going through the sample stream. Now, the cells are quite large. They essentially take up the width of the sample stream. So it's fairly difficult to get more than one cell to come through. However, if you like to run your sample on high, then generally this does allow that core stream to be wide enough to have multiple cells. But it would never be quite as extreme as if you have small particles coming through, because the small particles themselves, you could fit many, even in sort of the narrowest of core streams. So when you have multiple coming through, we call this coincidence, and coincidence can happen with cells as well. Now, in the extreme case of coincidence, we tend to use the term swarm for this as well. So what exactly does swarm look like in your data? We've seen it in this diagram, theoretically, what it should look like. And how can you avoid it, right? Most importantly, what does it look like and how do you avoid it? So Josh is going to go through some of this data from our paper using the recombinant EVs.
One of the easiest methods to determine the optimal concentration to run your sample at is by performing a serial dilution on your flow cytometer, because there's no optimal concentration across all flow cytometers, as they all have different laser geometries and they also have different core stream velocities. You'll see that when you do a serial dilution, that there should be a linear relationship between the dilution factor and the total number of events per second, or per unit time, or per unit volume. If you have a stable core stream velocity over time, then you can do it with time. If it's unstable, if you have something like a peristaltic pump, you might want to do it by volume. So the second thing with serial dilutions is that you should do it ideally with a stained control. It's possible to be detecting single particles, but if you have too much unbound antibody or too much fluorescence in the background, then your fluorescence signal won't be stable. So unlike the particle count, where it should be linear with dilution, your median fluorescent intensity should basically be stable with serial dilutions, because the fluorescence per particle should not be changing. If it is changing, it means that you are swarm detecting the fluorescence, whether that's unbound antibodies, membrane dyes, or something else.
So one of the easiest ways to determine whether you have unbound dyes or unbound antibodies is to run a buffer with the reagent alone at the same concentration that's being used for your EV or small particle samples. So something that we've learned along the way is not all reagents are made the same. Antibodies are generally nowadays QC'd and created for use with cells, and most companies do not QC those reagents for EVs. So it is very important for you to do your due diligence and check those reagents, because as you can see here, you can have a lot of particles just with the reagent alone, and as Josh was saying, that are fluorescent and can contribute to that fluorescent background. In this case, what you're seeing is separate concentrations of the same antibody. Now, this is probably one of the most extreme situations that I've seen. It was a homemade, home conjugated antibody with a conjugation kit that I would say is probably not recommended for small particle use, because in many times we are not washing our samples the way we do with our cells to remove the excess reagents. We are simply diluting. So if everything is in excess, then you will still see it, and that will contribute that to your fluorescence background.
And just to add to this, you can see that even when the optimal dilution is being acquired, there are still antibody aggregates that could be mistaken as fluorescently positive particles. And this is why a buffer with reagent alone is really critical as a control, even if you know that the sample is at the optimal concentration, because we don't want to report our data and then be unsure whether it was an EV or a virus you were detecting, or whether it was just antibody aggregates. And your reviewer will want to confirm that as well.
So moving on to the next step, we are now looking at flow cytometry data reporting and flow cytometry data sharing. These are very important in the reproducibility of your experiments and your data. Now this slide here, I am sure most of the audience cannot read this slide, and it shows to illustrate the importance of reporting in units or in a language that we all understand. So now when it's translated into English, you can now read the slide. It is about data reporting and how data transparency facilitates reproducibility. Data sharing to public repositories such as Nano Repository and Flow Repository for small particle data is important. So another aspect of data reporting is how you choose to interpret your data. How you choose to interpret your data shows through in how you report your data. And there are some market differences between the way cell data and small particle data should be reported, simply due to the nature of the type of particles that you are detecting. So what do we mean by this? When we're reporting flow cytometry data, we kind of want to highlight the importance of concentration instead of reporting in percentages. Why is this? So what you're looking at here is a typical example of what lymphocytes would look like in PBMCs. We are gating based on side scatter and forward scatter. These are characteristic populations that most people are familiar with if they work with PBMCs. These are the lymphocytes, which we just denoted with a circular gate. And we've gated the lymphocytes to then look at the CD3 and CD4 population. And now we've gated these guys, and it's about 53%. So what we're trying to say here, though, is that even with cells, T cells that we are very familiar with, these percentages can change depending on the placement of the gates. Now, if you were to change this lymphocyte gate to include this population, you will have extra events that come through in the subsequent gate and therefore change your CD4 positive T cell percentage. So gating in percentages can be inconsistent depending on how gates are being placed. Now, this is, of course, much easier to do in PBMCs and cellular populations where individual groups of events are well defined. So it is also dependent on having fully resolved populations. Now, when we're detecting cells on a flow cytometer, we never really question whether or not our cells are fully resolved by side scatter and forward scatter, especially when we're working with lymphocytes. However, we do have this problem when we're working with EVs. When we're working with EV data, I'm comparing now data that was collected on my cytometer off of side scatter versus forward scatter for recombinant EVs and versus side scatter and forward scatter for lymphocytes. So in this population, I do not have a resolved population in forward scatter. Side scatter, I do. So the distribution of events is very different. Many EV populations are not fully resolved the way cells are. And now I'm comparing buffer alone. So not only are we not fully resolving the population of interest, we have an overlap of buffer alone, which is the noise, over the population of interest. So you have background events that overlap with the particles that you care about resolving. Something that adds even more complication is the fact that buffer alone and dirty buffer can have a lot more events. So you're depending on what is in your buffer, not necessarily dirty. So if you've added proteins, if you've added any other particles in there for stabilization, etc., those particles will all contribute, and they're all in that overlapping space of where you expect to see your EVs. So keep in mind, if you're reporting in percentage, these events will all get counted into that percentage, and therefore make your percentages not as accurate as you would like. Hopefully that has given you some food for thought about reporting in percentages. And now we're going to move forward to talk about standard units. So hopefully what we have talked to you about earlier in our presentation has convinced you that standard units through calibration is the way forward. But this is just to demonstrate a little bit with some data for you to have a comparison. So on the left-hand side, you're seeing the PBMCs with CD3 staining versus side scatter. And on the right side, you're looking at these recombinant EVs that have GFP and arbitrary units. So these two populations are being shown in arbitrary units. And that is really just to highlight the fact that you cannot really tell if something that you are looking at is cells or EVs, which you know will have very few copies of that GFP on the surface simply because of the size of the particle. So just by looking at arbitrary units, it is very hard to say, yes, this sample is EVs and no, this sample is, not EVs just by looking at that fluorescence. But what you will be able to do though, is if you were to convert things into molecules of equivalent fluorophores, you will now know that it's not arbitrary and that you are actually dealing with very few copies of EVs. When this sample is converted to standard units of EV diameter versus FTSE MESF or ERF, we can then determine that the gates placed here are centered on a median fluorescence of about 290 FTSE ERF and with a median size of about 123 nanometers. Now, you might be saying, Vera, we would never draw gates like that. And this is just an example for you. But drawing a gate in a square actually is a very specific defined boundary for these EVs that I'm identifying. So the idea is to give almost like a GPS coordinate of how to find these cells again. So what I'm looking at is defining the exact boundaries of the gate by diameter, so 100 to 150 nanometers, and also by fluorescence, so 100 to 1000 FITC ERF. By doing so, I can give somebody the exact coordinates of how to find these EVs that I saw on my instrument later when they try to look for the same EVs. So because I ran these samples on a volumetric instrument, I know the exact volume that was acquired for this many numbers of EVs, and I can very easily calculate a concentration. So here, when we report with defined boundaries as well as standard units, it is truly a way to be able to tell people how to find something afterwards. It's like giving them a map, giving them GPS coordinates. So just to give you some context, I showed the CD3 T cells earlier. On CD3, or sorry, for T cells, you're expecting about 100,000 molecules of CD3. And if we assume that there are about 5 FITC molecules per IgG, that should be something around 500,000 molecules of FITC. So you truly do know that this is an EV population when its approximate fluorescence equivalent to 290 FITC MESF. So it's definitely not a cell. This brings us to the last of our tutorial objectives, advanced analysis. So here's where things get fun and interesting. Here's where we finally get to apply all these tools and methods to the biology that we all want to get into studying.
As Vera has spoken about, like calibration is really critical. Getting hardware agnostic data is super useful for you to take your flow cytometry data and go and compare it to something else like super resolution microscopy. We compared it to resistive pulse sensing and EM at the start because we had units of diameter, not light scatter in arbitrary units. The other thing with calibration is in terms of biology, we want to understand proteins, like how many proteins are on the surface of our particle and how does that abundance change over time, or how does the density of proteins change? And we're also going to get that from calibration. We can convert the number of molecules on an antibody and get an idea of the number of proteins that are stained on the surface. And then if we know the diameter, then we can derive the surface area of these spherical EVs and understand the density of the proteins on the surface. So if we go one step further with this, another characteristic that we've also spoken about that's important is refractive index. So if we know the diameter and we know the collection angle, which we get from our calibrations, we can also derive the refractive index of particles. And that's going to be useful in telling us about its structure, its morphology, whether it's a virus or whether it's a liposome, as they'll have very different refractive indices. So just finally, I want to mention a couple of useful resources. There's a huge amount of content to take away from this talk, but we've worked on a lot of manuscripts that go over all of this detail, so you can study it in far greater length and in your own time. So my FlowCyte EV is about the reporting requirements so that you can make sure that people can reproduce your work. The EV flow cytometry compendium is a great sleep aid. It will help you understand all of the complexities of fluorescence and light scatter and instruments, and also all of the controls involved with EV analysis. And then finally, MySev, if you're focused on EVs in particular, is more of an exhaustive guideline to help you look up how to report across lots of different pre-analytical variables like isolation and separation down to analysis methods. And then finally, if you go to the EV flow cytometry YouTube page, you can find a bunch of previous webinars that are focused on different aspects of EV flow cytometry.
just to add to this quantitative analysis of virus particles, the number of receptors on a virus, especially the receptors that mediate entry of a virus into a cell, is extremely biologically relevant. In this case, we're looking at anti-GFP, and the GFP happens to be associated with the glycoprotein, the envelope glycoprotein of this virus. So being able to know the number of copies, being able to quantitatively deduce sort of the approximate number of entry receptors for a virus will be extremely important for the biology and understanding the biology of these particles. So yeah, in terms of quantitative analysis, this is the next step for you to understand more the biology of the particles you are studying. And last but not least, we'd like to wrap up with the lessons learned. Size matters not. Size does matter. So lessons learned here, calibrated data should be arbitrary, they should not. Optimized detector settings use, default settings do not use. How do we get Yoda to say that? Jesus. Thank you very much for listening.
Thanks so much, Josh and Vera. That was a fantastic presentation. Very,
I'd forgotten we ended it like that.
very entertaining for sure. Do you guys want to answer the questions in the Q&A first, or would you like to go to your discussion slides?
I don't know, Josh, your call. You have the control of the slides.
All right, we'll go with some discussion questions.
Yeah, so I guess we've given this type of talk in quite a different range of venues and to different audiences, and we do kind of consistently get some common questions. So thought we would just summarize them. I think there's sometimes a certain level of confusion when it comes to the use of polystyrene beads. I think there was almost a stigma initially about people using polystyrene beads, that they were bad, because they don't represent the refractive index of EVs, and that's totally true. But in the case where you're utilizing them as though we are here for scatter calibration, they are actually a really great tool, especially the ones that featured here. Thermo Fisher sells them. They're NIST traceable sizing standards. And we do take into account in that calibration with the software the refractive index of the polystyrene, so that we can utilize that to calibrate your EVs. So that is a good use of polystyrene beads. Other situations where people are asking about fluorescent bead mixes, namely, I think there's been some popular products such as Megamix. Fluorophores we also use, but we don't use them for calibration. But Megamix beads, we cannot utilize fluorescent beads for light scatter calibration, because the fluorescence will also interact with light and it will not give you, and these are also not NIST traceable. So the sizing of these beads is not super accurate. And I guess the last but not least, we sort of touched on it a little bit. The last number 3, I'm seeing a lot of it being published in research now, where people are just kind of drawing a gate to determine if something is their EV population based on a mix of beads that they're running. So Josh went over this, I think, quickly during the talk. I don't know, Josh, if you want to add a little bit more about why it's not a great thing to do to set your gates based on beads.
Yeah, I guess just the main takeaway is if you run 100 nanometer beads and 200 nanometer polystyrene beads, and then you go between
a few different flow cytometers, basically the distance between that 100 nanometer population and the 200 nanometer population, the ratio of those populations will change depending on the collection angle. So unless you do light scatter modeling, there is no way to use light scatter as a standardized parameter to record concentration between flow cytometers. It's the only way that you can make it hardware agnostic, as we keep mentioning.
Yeah. So if you buy a mixture of beads and you draw your gates based on this population to this population, somebody else on a different cytometer can buy the same beads and draw that same gate, and it would actually be a different, slightly different population that they're gating on, right? Kind of what we're trying to say.
Yes. And then sorting.
Joanne, you want to
favorite
take this
subject.
one? We've just been getting a lot of questions about sorting. I think that's sort of the natural progression of the field, where people are like, hey, we're seeing these particles now and we want to sort them. But there are a lot of caveats. So I don't know. Josh, you want to start? You've done sorting. You've actually done EV sorting. Like, you were part of that crew sitting there.
So the main thing to take away from sorting EVs is sorters, the commercially available flow cytometry sorters that are exist were not designed to sort nanoparticles. They were designed to sort cells, and cells, most cells that are aimed to sort are kind of 6 to 10 microns, give or take. So the droplet size itself, even if you use a small nozzle, that droplet is going to be the difference in size between like 100 nanometer EV and the droplet is kind of the difference between 1 nanoliter and a liter for every single particle you sort. So what it means is that most of our analysis techniques, if you want to do downstream validation of what you've sorted, and you go to NTA or resistive pulse sensing, where you need 1 times 10 to the 8 or 1 times 10 to the 9 per mil, you will need to sort liters of EVs, which means that you will be there for an extremely long time. It means that you will also need to concentrate, because you basically have a 10 to the 9 difference between a single particle and the volume that it's diluted in. And then when we concentrate, we lose particles. It's just not a very practical solution, even if it worked perfectly. And then the flip side to that is we keep speaking about limit of detection. So most of the sorters that we have today, I imagine the most sensitive can probably detect 80, 90 nanometer EVs, if we're being kind of generous. The majority of EVs are still below that. Most EVs tend to have a modal point at kind of 50, 60 nanometers, and they're log normal. So if you sort a particle, it's very possible that there are 10 more particles that are smaller in that space, or background debris. We spoke about antibodies. There's still going to be stuff in the background. It's not going to be a pure population. It may be possible to sort subsets, and if you plan to just go and reanalyze by flow cytometry, that's maybe feasible. But downstream assays where you need very high concentrations, with our transfection assay that we showed in a manuscript done at NIH just as I was joining, they sorted for 48 hours straight to get enough material to do a transfection assay. So it's extremely resource intensive, and if you're working from a core, it's probably not possible to hire your instrument for 48 hours unless you're allowed to be extremely disruptive to other users. So I don't know if Vera or Joanne has anything they'd like to add to that.
Joanne?
No, I think those are all very good points. And, I just warn people that tell me all the time they're convinced that they have a pure population because they rerun it through the same instrument with the same limit of detection, and they see a pure population. It's what you can't see that is concerning. And just like we know with cells that if we set our threshold too high to get rid of debris and red cells and things and platelets like that, that the sorter can no longer see those, and they can't make a sort decision, and those particles randomly end up in your sample. So the same principles apply here, but the only thing is it's not what you're setting your threshold at. You're probably at as low as you get, but there's still stuff that's not going to exceed that threshold.
I guess sorting in this case is always going to be an exercise of enrichment and not
Yes.
really finding a pure population.
Yeah, and you have to be careful about what attributes you give your sorted cells in your downstream applications. If it's a functional thing, you have to be sure that the number of EVs that you think you're sorting are the dominant population and are what the function is being attributed to.
And I think just to add to it, I think sorting can be pretty feasible for cell culture where you have relatively pure populations and you can use FBS-free media. If you plan to sort whole blood or plasma, serum, where there is tons of lipoproteins and other macromolecular complexes, that's where it starts to get fairly infeasible to get a pure population because there is just so much stuff that is below the limit of detection that will still be in those droplets.
Yep.
Yeah. And I think practically speaking, I feel a lot of the cytometers where we're doing the analysis on EVs and getting like very, very small, a lot of them are using water, like ultra pure water as the sheath. When you're sorting, you have to go with some sort of PBS or isotonic solution, right? Because you don't want to lyse all the particles that come out afterwards. And that presents, I think, more challenge in terms of adding more noise to the system that then you have to figure out how to remove. So it really eats in on that limit of detection that you are able to get on an analyzer, but maybe practically speaking is not going to be so feasible on a sorter.
Yep. All right, do you guys want to go to some of the questions that have been posted here?
Yeah, we didn't
Um,
get as many as I thought we would.
Oh, they'll start coming in as people think. So the first one that came in says, what rumors surrounding instrument setup would you like to set straight? That's a loaded
First of
question.
all, what rumors do we know? What are some rumors that we? I mean,
Well,
I think we
there's
sort
a
of
rumor is that you set your instrument settings to get a certain size particle on scale and you're good to go.
yes. I mean, what?
It's hard to know where to start. I think the main thing you should take away is that default settings are never optimal for small particles. They are designed for cells, and they are also, day-to-day assay settings are designed to keep a system stable. They're not designed to optimize to get the maximum sensitivity day to day. So if you want to look at small particles on your instrument, you will need to optimize your settings, and that's the main reason that we showed this pipeline. And then the only other rumors really about how you interpret the beads. So all of the ways that we've interpreted beads here is we don't take them at face value. We've used polystyrene beads and we've done modeling and accounted for that. Our Earth, like broad spectrum beads that are used to set up flow cytometers that are usually QC beads. We have personally cross-calibrated them with MESF beads to make them useful for our instrument and to optimize our data. And then the area to height thing is, I think, we showed fairly convincingly that it's the only reason you would get a better height signal is if you haven't optimized your window extensions. And area is going to be more robust for you to do calibrations down to the limit of detection where it's the most problematic and you see the biggest divergence between the two statistics.
Great. Next question. Oh, did you want to say something else, Vera?
Well, I was just saying about how there's so many people who do that. They put a bead on and they're like, hey, I can detect it and therefore I'm good. I guess it's one way to do that. If you're going to do that, please calibrate so that you know what you're detecting. But the other thing is you'll never know what is the most sensitive your instrument can be if you do it that way, right? Because you're just finding a setting that will detect a bead and then you're good to go. So yeah, just keep that in mind if that's the method that you are using.
And just also keep in mind if you're detecting 100 nanometer polystyrene bead doesn't mean you're going to be detecting 100 nanometer EVs. So if you're setting it to that size particle. Okay, next question. Does this side scatter sensitivity announced by manufacturer play a role? For example, in BD instruments, the side scatter sensitivity is usually reported as 0.5 microns. Can we get smaller particles by optimizing?
Oh, I've done that on the Vortessa.
Yeah,
That's
the thing to keep in mind with manufacturers is the spec that they put in their technical specifications has to be valid for all of their instruments and passing QC. It's very possible that well, by default, all instruments will be able to reach what they put in their technical specifications, and likely all will go beyond it, but to varying degrees. So you absolutely can optimize to smaller than technical specifications. It's just manufacturers aren't going to be held accountable if one instrument can go to 100 nanometers and another one's 200 or 300 nanometers because they said all of them can reach 500 nanometers.
It was always the worst, the weakest link is the is what is published.
Yeah.
Yeah, that's probably the worst case scenario.
Worst case scenario.
There are some posts about running EVs on high. Can you comment on why this is not recommended?
So I guess that question came up, I think, before we show the video was at the part about coincidence and sort of the width of the core stream, what it means on high. So I mean, when you run your sample on high, you're essentially opening up that core stream so you can have any number of your EV particles coming through. So it's not recommended because you would like to have a single particle UV come through. And I guess like, I mean, what?
To give like more of a technical answer for why you shouldn't do this is, as Vera said, your likelihood to increase coincidence detection is increased, and you'll probably hear a rebuttal that says, well, I can dilute my sample more, and then run at the same concentration, a lower concentration that accounts for that wider core stream. The reason that you don't want to do that is when you are analyzing small particles, you want to keep the CVs down and you want to keep the signal to noise as consistent as possible. When particles run through a core stream, they don't actually all run down at the same velocity. There's kind of like a U shape, and it will increase and decrease around the sides. So if particles spend longer or shorter in the core stream, their illumination will be different and you'll get different CVs. So you'll increase, if you widen the core stream and detect over a wider period, you're going to get more variation in the signal, just by proxy of the variation in the core stream velocity. The second reason is that a lot of laser beams are either Gaussian or top hat. And the truth is that even the top hat lasers that are meant to be relatively stable are actually quite messy at the top. They're ragged and they're not perfectly flat. So again, if you increase the core stream diameter, you're varying where EVs intercept that kind of raggedy edge. And again, that will change your CV. So you want to keep your signal as consistent as possible to get the highest precision. And that's why having a smaller core stream is going to help you do that. And the question of coincidence ultimately does come down to the dilution factor.
Yeah, just one more thing to add. I mean, the argument that I've seen people present is that I get a higher number of fluorescent events in high. And the thing to think about that is, are you really getting a higher number of events, or are you having multiple very low intensity events count as one single brighter event that's now exceeding your limit of detection? So it may not, it could be definitely artifactual, these increases in numbers of fluorescent events.
And your zero dilution should elucidate that for you if you're unsure.
So here we are back to sorting again. I don't know if we've answered this person's question or discussion about sorting, but I'll put it out there again. Do you have recommendations when trying to sort EVs in terms of nozzle size, pressure, or even the efficacy of the process?
Yeah, I think we've answered that. And as we
Yeah, I
said,
think the only
nozzles.
thing, right, nozzle size, I think I would, you have to consider the smaller the droplet, the better.
But I feel like So in my experience way back with the Astrios, our smaller nozzle was always noisier. So we didn't
That's
put
the other problem. Yeah. Because your frequency is higher.
yeah. So it actually ended up we defaulted back to the 100 micron.
Yeah, that'll
So
help with your noise. But also, if your pressure is lower, then you have less dwell time in the laser and you're getting less photon generation. I mean, sorting is a challenge only for those who like self-abuse, in my opinion. Okay, you are using polystyrene beads to set size. Are you measuring EVs in absolute size or relative size?
I'm not sure the intended definition distinction between those. The size that's being measured is the derived size based on the light scatter model. So depending on whether you use a core shell model or homogeneous low refractive index model, um, it's a size derivation. It's neither absolute nor relative. But the polystyrene beads are only acting as a calibrator to derive the model. Once the model's there, it's set, and everything else is derived from the model, and it's nothing to do with polystyrene beads anymore.
Okay. Thoughts on apogee beads?
My experience with apogee beads is they can be the refractive indices differ a little bit when you start to model them because they're silica, and you even see that they actually have different silica refractive indices in their technical specifications. I've also found that they've just degraded a little bit faster and they're less stable. And they're also a lower concentration. So if you buy some NIST traceable beads, they're probably going to last your lab a lifetime, and they'll be stable over that lifetime. Is my personal opinion. It's not something I've published or.
So how do you feel about them being something that is run after the instrument's been optimized as like a daily sample to verify that the instrument is still optimized?
That's certainly one use case for them. Again, if you're going to run them as a control, a positive control, and you want to use it longitudinally, I think the degradation for me is personally a problem. And any type of bead that gives you a positive signal can be a positive control.
Right.
It's
Okay.
hard to use them for if you were thinking about using them for light scatter calibration.
Right.
It's not
Yeah.
recommended.
Well, this is an interesting one. Please dispel laser wavelength and side scatter sensitivity.
Oh, Evan.
The main in physics, if you have a lower wavelength, there'll be more energy for light scatter. The problem is when it comes to a flow cytometer, there may be more energy of light scattered, but it's how that energy is collected and actually taken to the detector. So at the low end, detectors sometimes aren't as efficient down at 400 nanometers, nor are the collection optics. You get chromatic aberration. Most flow cytometers have a relatively simple collection lens, and the focal point when you have a pinhole and you're trying to focus the light on that pinhole, you'll get concentric rings where all of the light goes to slightly different points, and they'll be more fuzzy or not. Most flow cytometers are going to align, to the mid wavelengths because they're collecting from UV all of the way up to the far reds. And so if you have side scatter at 405, which a lot of manufacturers have done, if you really try and optimize that, if you get an engineer to come in and peak that detector and peak um, the optical fibers, you'll actually find that all of your far reds now have lost sensitivity. And this is all of the interplay with the optics of flow cytometers. So you could make them best, but you'll trade off fluorescent sensitivity, or you could look at like a 488 nanometer light scatter. The other thing is that light scatter, the amount of light scattered from a particle is dependent on its composition. So polystyrene and the amount of the refractive index changes with wavelength. So what happens is at 488 and you go down to 405 and the refractive index actually increases. So you get this curve upwards. That curve is actually a lot steeper for polystyrene than it is for silica and for EVs, where it stays a little bit flatter. So actually, most manufacturers will look at a polystyrene bead to test all of their sensitivity and validate it. What they're not looking at are the silica and biological particles, and there's actually a bigger difference at 405 than there is at 488. So if you can detect 100 nanometers at 405 and you had the same detection sensitivity at 488, strictly speaking, you would be more sensitive for biological particles at 488 than you would at 405, because there's a smaller difference between the refractive index of polystyrene and vesicles at 488 than there is at 405. So hopefully that made sense and wasn't too much gobbledygook.
Can you comment on laser power to make up for a difference in refractive index response?
Yeah, laser powers, I mean, most flow cytometers will still be blocking light. Even with small particles, they're not collecting 100% of light. And as you increase laser power, you also increase noise. So there's a fine balance between the two. And actually, most of the most sensitive flow cytometers on the market actually use really low laser powers to decrease noise, and they just have really sensitive detection optics. The other thing that you really need to think about is when you increase laser power, fluorescence isn't, it's linear to a point, and then you will start photo bleaching your fluorophores. And scatter's great, we need to detect particles, but ultimately the reason we're using flow cytometers is because we want to see what proteins are on those particles. So if you completely deprecate your fluorescence sensitivity just to gain a little bit more scatter, there's not really a benefit there.
Great, thanks. Oh, I think you should answer this one, Vera. What is the refractive index of metachlorian?
I have absolutely no idea. I feel like we could try that experiment.
Thanks, Derek, for the chuckle.
Yeah, I'll see you in
Do you
January. Sorry, in February.
Do you have some recommendations for the Cytoflex Nano or reducing background when you look at particles as small as 40 to 60 nanometers?
That's a very, very specific question, because background could come from so many things, right? I feel like
I think that's a offline question.
Yeah, in the nature
Yeah.
of preserving the agnostic aspect of this webinar. Yeah.
Okay.
I mean,
right.
there's a lot of different things that can come into background. So, you know, like your own buffers, and we talked about the reagents that are in there, the salts and the buffers and that sort of stuff. So,
Well, also remember the core size is also going to contribute to the amount of background noise you get. The larger your core size, the more surrounding material around your particles that is going to generate optical noise. So core size is also important. Would the imaging capability of a cell sorter be helpful to get a cell sorter more capable and more sensitive for small particles? We have experience in using ImageStream in the past.
this is a Joanne question.
Yeah, it's a depends answer. It really depends on the sensitivity of the imaging system that you're using. I think, I'm not sure that any of the sorter or sorters have that quite sensitivity in terms of imaging yet, but it's yet to be demonstrated, or I'm not even sure if it's been evaluated. In theory, imaging tends to be more sensitive for smaller particles. But again, there's a lot of criteria that would have to play into what the actual imaging detection system was. Would you agree, Josh?
Uh,
I mean, there are
I
single molecule imaging systems, right? But those are not going to be on sorters.
I
yeah,
mean, you work for the company that has the only imaging cell sorter right now.
yes, and that technology is not the same as the image stream, and it's not going to help you with sorting smaller particles.
Not to say that sometime in the future we won't have those tools for sure. Have you ever tried sorting on a Tyto?
I have not.
No.
I haven't either. Okay. Um, It says I'm guessing, have a BD custom kit to detect EVs, and they recommend using true count, absolute counting beads in order to evaluate concentration. Is this a problem? Do true count beads interfere?
I imagine that's because the BD instruments on the market right now don't have an inline flow sensor. So to derive concentration, you'll need a spiking of beads. Um,
So
so
I mean, I do have a little bit of experience using true count beads. We've bought them before. My problem with them, if I recall correctly, is they add a lot of background. The true count is the one that's the little pellet on the bottom. And I think that pellet formulation, when it comes to cells, you don't see because they're like, you're not looking there. But if you're down at the EV level, it actually contributes a fair amount of background because I'm guessing you're spiking your EV samples. The way you use them with cells is you put your cell sample with the beads, and that formulation of the pellet in my hands was contributing a lot of background noise to the sample.
Yeah, the other thing is to consider is the size too, and
They're huge.
whether or not these very large true count beads are going to contribute any interference in terms of signal due to their size. I guess you would have to look at them by fluorescence and not scatter. They'd be off scale for scatter,
I mean, you could set your forward scatter for the true counts and
I
then,
guess. Yeah.
but yeah, that was my experience noise.
Any thoughts on Celarcus kits with use on spectral cytometers to detect EVs or viral particles?
Once again.
um, I think the Solarcus kit is based around a membrane dye. Um, the premise of that is the amount of dye intercalated into the particle is proportional to its total surface area. And then we're assuming that all the particles are spherical, and then you can basically derive the diameter from a surface area. So you can validate this for yourself, especially on a spectral flow cytometer. If you have a standard, you need to, if you're going to use that standard to calibrate your particles, you need to detect the whole standard. So can you detect the whole standard to begin with, in order to do that calibration? And then all of this is based on the intensity of light between that standard and that standard size, and then the particle of interest. So is the spectra the same? Or is it changing between the particles that a membrane dye is intercalated into? And with that, um, with your particles, if you have particles that are more proteinaceous or less proteinaceous, like a virus compared to an EV, do you expect the membrane dye to intercalate into that particle the same as it would a liposome that doesn't have any proteins or complexes on it blocking the intercalation of a membrane dye? So the reproducibility of it, and the specificity of it, is something that you should validate for your samples, just taking into account the first principles of how the kit works. And that's the same with, anything you do, you should kind of take into account the first principles and say,
Yeah.
how does that apply to my assay?
I guess it also depends on what your goals are for if you were wanting a fluorescent standard unit to report to, or you're actually looking to size the particles, right? Because like Josh is saying, the size is related to the amount of dye, and the amount of dye is related to the biology of the particle you're looking at. So to have an accurate measure of size, the biology of what you're looking at needs to reflect the biology of the standard that's being used. But if you're just talking about reporting in a fluorescence unit, like a calibrated fluorescence unit, I guess, I mean, that still would work, right, Josh? Because they would all have a certain level of fluorescence and they would be in sort of the realm of the size that you're looking for in terms of EVs or viruses, right? But if we're looking for an accurate measure of size, that would depend on how that dye behaves with the biology of the particle.
Yeah,
I believe the kit uses the antibody binding beads for calibrating fluorescence. Is that not correct?
Okay, so then it's the
yes, but you would, but it includes the liposome standard where that should validate those beads. So as we spoke about, when you calibrate on someone else's flow cytometer and then try and do it to your own, those are two different systems and they have different tolerances, and the way that a broad spectrum dye works or will interact with that difference compared to the dye you're actually interested in are different. So you should always aim to validate. The other thing is that membrane dyes in particular are something that everyone wants, a universal membrane dye that works across all of the EV derivations and sources. I personally haven't found one, and I've screened a lot of dyes. You'll see that there are quite a few manuscripts now in Jev and other places that had people take all of the membrane dyes and compare them all. And there just isn't a best for everything. They seem to be selective based on the lipid composition, which isn't surprising. And the properties of some of these dyes actually change as well, depending on the lipid that they are intercalated into. It can change their spectra. So some people actually use these dyes as reporter dyes for cells to know about the lipid composition. Staining is one thing to get a positive signal. Deriving size from anything, whether it's light scatter or fluorescence, requires assumptions, and you just need to make sure that those assumptions are valid for your assay.
Okay, we're getting close to the end of our time, or we're actually at our end of our time. So let's go to one last question. So Cynthia doesn't feel left out. Cynthia wants to know what you think about compensation in multiparametric EV experiments.
The main thing to keep in mind is every time you do compensation or spectral unmixing is you have errors. You get spectral spillover, and when you have errors or you need to compensate, it basically means that you are lowering the limit of detection in whatever channel that is compensating. So if you're really doing EV flow cytometry, single EV flow cytometry, you need as much sensitivity as you can get to phenotype a lot of this really small stuff. And that usually means that you have to have at most a dye per laser and ideally have almost no spectral spillover, in which case compensation should be an issue. If you do have significant compensation, you're deprecating the sensitivity of your assay to actually detect the things that you want to look at. The other thing to add to this is that multiparametric EV experiments by nature are very difficult just because the antibodies, are kind of 5 by 15 microns, nanometers, roughly. You start getting steric hindrance if you try and put too many different types of antibodies on the surface of an EV, and you'll notice that the order of the staining can change the output. So in general, I wouldn't recommend doing too many colors or not even needing to compensate entirely.
I would agree. Okay, I think it's probably a good time to end our webinar. On behalf of the ISAC community, we'd like to thank everyone who joined our presentation today. If you're interested in participating in the other incredible learning opportunities CytoU offers, please check out our archived Cyto recordings and on-demand webinars. We greatly encourage viewers to participate in the online survey once the webinar is concluded. Your feedback is highly valued in advancing our educational program. This webinar is also recorded and will be available to everyone. Please keep an eye out for more webinars in the future. Until then, have a great day, and thank you, Josh and Vera, for an outstanding webinar and discussion. Have a good day, everyone.